So once again, good morning or good afternoon, everyone. Welcome to today's Catalyze Investigator Panel webinar. My name is Patrick Ahern with RTI, and I'll be helping out with the virtual room today. So just a quick note, when you log in, your microphone will be automatically muted. We are recording today's session, and if you have any questions for our panelists, you may submit those into the Q&A window at any time, and we'll try to address as many as possible during today's session. Closed captioning is available during today's webinar. You may turn those on by clicking the Show Captions button on your main Zoom toolbar. If you run into any technical issues, please let me know in the chat window. So at this time, I'll go ahead and turn the meeting over to Marissa to introduce today's panel. Thank you.
Thanks so much, Patrick, and hi, everyone. Welcome. We're so glad that you're joining us today for what is sure to be a fantastic webinar. My name is Marissa. Hopefully I'm a familiar face to many of you. I work for RTI International on the side of the Catalyze Coordinating Center. I host our skills development program, so things like our webinar today I've put together, and I'm happy to see so many familiar names joining. So for our webinar, we have 4 funded investigators who will describe their Catalyze project and summarize key takeaways from the program, giving you a firsthand look into some of the awesome technologies that we support, as well as what you can expect from this unique program as a Catalyze awardee. It is a unique perspective to hear directly from the investigators themselves, so I'm really happy to have the opportunity to host these 4. And keep in mind, we will have time to answer your questions at the end. So please, like Patrick said, be thinking of your questions and type them in the question and answer box for us. We will get to as many of those as possible. So our first presenter today is Dr. Rebekah Gundry from the University of Nebraska. Dr. Gundry was part of one of our first cohorts of Catalyze awardees. So she's really grown with the Catalyze program, as has her technology, which is a research platform. So without giving too much away, I will turn it over to Dr. Gundry.
Oh, and you can see my screen okay? Yeah, oh, great. So I'm Rebekah Gundry in a blizzard in Omaha right now, and my background is I have a degree in forensic science, and then for my PhD and postdoc focused on biological mass spectrometry and its application to cardiovascular disease. And really the driving inspiration for the product that I'm going to tell you about is in part my absolute fascination with the power of technology for improving human health, along with the biomedical need. So our product is GlyPack, which is the first step in a turnkey glycan analysis platform that breaks through, I got it, hungry cat, sorry, through the traditional barriers of glycan analysis. And so with GlyPack, we're changing the future of biomarker discovery to develop safe and more effective therapeutics and more accurate diagnoses. And so the motivation to develop GlyPack is very simple. 93% of human diseases lack suitable biomarkers for diagnosis, prognosis, and treatment. And without good biomarkers, disease diagnosis can be delayed, and treatment commonly proceeds by trial and error. So a solution to this problem is to exploit the sugars, or the glycans, which coat every cell type in your body. So they coat every cell type, antibodies, and viruses. They're on red blood cells, and the glycans on these red blood cells define your blood type. And as an example of how glycans are essential to life, if you receive the wrong blood type in a transfusion, you can die. So incorrect glycans are present actually in every major disease, making them an excellent source of biomarkers. However, there are major challenges to analyzing glycans in the laboratory. So GlyPack overcomes several glycan analysis challenges by being easy to use. It's capable of preparing 384 samples in a single day, starting with about as much starting material as you would find in a grain of salt. And in one example, results from GlyPack clearly separate out patients with rheumatoid arthritis before they receive treatment. And the 2 groups here are before the first treatment, patients that ended up being able to respond to first-line therapy versus those who did not. And this means that doctors could immediately start the latter group, the ones who are known not to respond to the drug, on a more advanced therapy right away, saving health and money if we are able to implement this in the clinic, for example. And so I'm proud to say that our product GliPack has been licensed through this Catalyze process, and it's being produced and sold by a company called Protify. And so the impact of Catalyze for me was that kind of the highlights are that I got regular interaction with folks that I wouldn't normally interact with. So NIH program officers and other NIH scientists and staff, legal experts, business developers that overall gave me access to expertise and experiences beyond what I could access locally in my own institution. This was really big for me. I also received a lot of input on target markets well beyond what I had originally anticipated when I was writing the award and just thinking about the application of the technology. Also having the quarterly meetings, this is very, this is certainly different than your standard NIH grant, but meeting with these folks on a quarterly basis was really helpful to keeping the momentum going. And also with, having it, a routine metric of accountability, it also helped us address challenges really, really early in getting help right away. I was also provided an opportunity through this program to attend a venture capitalist meeting where I could see examples of other startup companies, see how they're pitching their products or their solutions. I learned a lot about what works, and I also saw a lot of not great presentations. So it really helped me to identify some of those things to avoid when developing pitches. Certainly, I learned a lot about different insight about what investors are looking for, which is certainly a different way to think than I would normally do in kind of my day-to-day academic job. And then in terms of entrepreneur development, this was huge for me. At the start of this process, I had zero experience. I did not think about commercialization of anything at all. I found it to be very intimidating. Science and academia has its own challenges, and I think I, really kind of got stuck in that. So it really forced me to learn aspects of the business side that I'd never thought about before. And then at the end of it, of course, with all of the guidance and the practice and with the pitch creation and just the overall process of how things are developed in the laboratory, moved to industry. It's, I'm certainly obviously not perfect at it, but I have a much better idea of what to expect and how to approach it. And it still is impactful to me to this day, because now I'm actually in the very early stages of forming a startup company around another one of our technology platforms. And so this is something that I don't think I really would have even entertained if I didn't have the confidence and the experience that I got with the Catalyze program. So I will stop there and look forward to questions.
Fabulous. Thank you so much, Dr. Gundry. As we said, everyone, please, if you have any questions for our investigators or our panelists in general, drop them into the question and answer box, and we'll take them at the end of our 4 investigator presentations. So next, I'm happy to welcome Dr. Benza. He is a current product definition awardee who is developing a medical device at Mount Sinai.
It's great to meet everyone today. I'm going to be talking a little bit about my product development, which is the development of a biatrial catheter for cardiopulmonary support in patients with pulmonary hypertension. So a little bit about myself. I am been an academic cardiologist for 30 years, and my specialty is advanced heart failure, cardiac transplantation, mechanical circulatory support, and pulmonary hypertension. I'm currently a professor of medicine at Mount Sinai here in New York, and my therapeutic and research passion is in pulmonary vascular diseases, both in terms of developing risk stratification tools and in drug and device development for this disease state. So the particular clinical problem that my catheter addresses is one that many of our patients with pulmonary vascular disease have in their course of their disease state. Many of my patients have severe pulmonary hypertension, meaning a very high pressure in the pulmonary arterioles, which lead to an inordinate stress on the right side of the heart, causing right heart failure. And that's how many of my patients die from this disease. And once they develop right heart failure from their pulmonary hypertension, their risk of dying is in excess of 70%. Now, in a disease state that affects primarily middle-aged women, this type of attrition rate is just unacceptable. Now, we have pharmacological therapies that are designed to treat patients with pulmonary hypertension, but they only indirectly help the right heart by mitigating the pressure in the lung. They don't do anything to the right heart itself to support it. And unfortunately, there are no mechanical devices or artificial circuits that currently have to offload the failing right heart to allow our drugs to fight the disease and stabilize patients or allow them adequate time to wait for lung transplantation, which is really the only cure for this disease. Our product solves this dilemma by developing a specialized catheter that allows us to decompress the right heart, to restore circulation to normal, allowing our medicines time to combat the disease, or allowing our patients to stabilize and wait for lung transplantation. So our solution to this problem, we sought to design and prototype a new catheter that could radically improve patient outcomes with patients on extracorporeal membrane oxygenation. This is an artificial circulatory circuit that we use for patients with very advanced heart failure. And our current catheter eliminates many of the current related problems that we see with the use of these standard ECMO circuits. Our product is delivered through the skin, and it's deployed in a vein in the shoulder and neck. And this is very different than the standard ECMO circuits, as I'll show you a little bit later, which are deployed through arteries in the lower extremities, both compromising their peripheral circulation and also prohibiting them from walking around and rehabilitating themselves. It has a dual-lumen venous design that avoids injury to the arteries and prevents limb loss. And it involves a transatrial septal approach. So instead of a catheter that removes blood from the right side of the heart and deliver it to the left arterial system, we go directly from the right atrium to the left atrium. And this does a lot of things that are helpful for the patients. It decompresses the pulmonary circulation, preventing further damage to the pulmonary arteries. It prevents the watershed activities that we have that results in underperfusion of the heart and brain. So just remember, when you remove blood from the body and then deliver it through a peripheral artery, it's going retrograde against normal circulation. And so this prohibits the adequate oxygenation to the upper extremities and the heart and the brain itself that can lead to a lot of problems. This catheter position really facilitates a stable position because we have a novel locking device that allows our patients to get up and ambulate and really extends the longevity of the system, allowing for discharge and further treatment at home. And our eventual hope is to facilitate a future mobile ECMO design built around this very special catheter. And so this is kind of the catheter design that we've developed. It's a dual-lumen catheter. And you can see on the upper portion here, the inlet catheter and then the delivery portion of the catheter. We have lumens that are developed within the catheter that allows both the inflation of our docking system, which is this balloons, which goes on either side of the interatrial septum, and also very specialized sensors that go on the right atrial side of the balloon to make sure we're adequately decompressing the right atrium, and on the left atrial side of the balloon to make sure we're not overfilling the left side of the heart too quickly. This is designed with a special diffuser tip that allows blood to egress the whole dimension of the left atrium and really kind of prevents the jet effect that many of the catheters have, which directly impinge on the atrial walls and can cause arrhythmias. So by diffusing the blood flow into left atrium, we get a much better natural flow and don't have the complications of the arrhythmias. So we're able to design this prototype again with many suggestions from our Catalyze program officers. And this is the benchtop system that we designed. We developed an artificial circuit in which we can deploy our catheter. And this is an example of the catheter going across the mock atrial septum. And we can actually do pressure circuits using this to make sure that the catheter alleviates the pressure on the right side of the heart and delivers the adequate pressure to the left side of the heart. And we were able to move from our prototyping on the benchtop into an actual animal model, which we're currently on in our project development, doing actual ECMO circuits on animals that have developed pulmonary hypertension artificially and have right heart failure. And this is just an example of the catheter with the two balloons deployed across the atrial septum, which is demarcated by this wire, and effectively supported this animal for 4 hours. Now, as I mentioned before, this is how most of my patients look like who have ECMO. They're lying flat on their back. They have tons of machinery around them and big catheters in the leg. And you can see the size of these catheters, and you can just imagine how they damage the arteries in the legs and could lead to limb loss, which is just catastrophic for our patients. So, oops, sorry about that. Our new design, I lost my little picture here, but maybe I can get that back because that's kind of the cool part of it. You can see it's going to disappear, unfortunately, but our catheter goes through a neck, allowing the patients to stand up and walk around with these catheters in the ICUs. And eventually we'll design our catheter, which comes again out of the neck, to a portable backpack system to allow these patients to have ECMO in the ambulatory position. The commercialization project has been fascinating to me, as Rebekah mentioned. Being a clinician, I was totally ignorant of this form, this side of medicine. But the commercialization process, developing a patent, getting a patent through, the development stage of developing a prototype, working with companies who actually manufacture the devices and deploy all the special bells and whistles associated with the catheter was a fantastic learning experience. Learning how to go from a benchtop to a large animal design was, again, something that that really was facilitated by participating in this grant award. And then particularly, working through the regulatory and reimbursement strategies, again, things very foreign to us, but now we have an existing CPT code for this catheter, and we'll be able to file our 5110 with the FDA already. And so the impact of Catalyze on this project has been really quite amazing. And I really want to stress this to all the awardees who are on the audience. The importance of this system to my project has been incredible. And I just wanted to highlight some of the things that I thought were particularly advantageous. It really introduces a unique interface between science and business. And like Rebekah said, nothing that we have ever, as scientists and clinicians, have ever really embarked upon before. What I thought were particular highlights are the seminars. So when you become engaged in the program, Catalyze has a number of seminars that works through each one of these steps. And I highly encourage you to participate in these seminars, because that's where I learned a lot of the nuances and the branch points to take myself into the development of my company and bringing it through the regulatory process. So please go to those seminars. The quarterly meetings, as Rebekah mentioned, are key. These meetings keep you on track. They're very engaging. You're just not reporting. It's a two-way conversation. It's another set of eyes on your projects. And my PO is particularly helpful in actually recognizing certain things that I didn't think of initially to help me to improve the project. So another set of eyes is critically important for each of the steps that you go through in bringing your product from a product to the marketing, commercialization, regulatory processes. It takes blocks and helps you mitigate them. So if you run into a stumbling block, there's active mitigation for that stumbling block in those sessions. What we can do, what other seminars we might be able to approach, different ways of approaching and getting rid of those roadblocks. So that's really critical. And obviously they continually to tell you about resources that are available through the program to help you mitigate these roadblocks. So these quarterly meetings are key. One of the big things that I really took out of this meeting is learning these transition branch points, from product development to the regulatory stage, and from the regulatory stage to the commercialization phase. This commercialization phase is particularly important in helping us understand the market for our products and to develop partner engagement versus independent development of our products. So how you offload your product at the end, whether you want to take it through full development, or do you want to develop a partner who will then take your product and bring it to full development for you? It really makes you a much more knowledgeable entrepreneur. So please actively engage in everything that this program offers you, and you develop a sense of comfort, like Rebekah says, that will help you make the next steps. And these branch point learning processes were particularly important to me. I hope that was helpful to everyone, and I look forward to the question and answer session for everyone.
Great. Thank you so much, Dr. Benza, and thank you for your plug for the webinar series. We definitely really appreciate that. Next, we have Dr. Raval, who is a preclinical investigator in the Catalyze program, who is developing a biologic at the University of Wisconsin. So, Dr. Raval, take it away.
Thank you. Can you see my slides there, I assume?
Yes,
Yep.
I can.
Very good. Okay, so about me and why I founded a startup company very quickly. So I'm the board chair and co-founder of Cellular Logistics. That's my email. I learned this from Catalyze, that I can no longer use my academic email. So that's one big point. You get a corporate email, it's important. And then of course I have a second hat at the University of Wisconsin. I chair the cardiovascular regenerative medicine focus group at University of Wisconsin, and the hospital arm of that is UW Health. So I've been working on cell-based therapies, regenerative medicine, for over 20 years. Dr. Schmuck was a postdoc whose mentor was retiring, and he was just starting, and he kind of joined my lab, kind of inherited him. And he had been working on something unrelated, mitochondria, and he was growing up fibroblasts and harvesting their mitochondria and stuff. And he noticed that in the culture conditions that an extracellular matrix was developing, kind of frothing to the surface. And so he came to me, and I knew nothing about mitochondria, and I said, well, what do you want to do with this? And he said, well, let's explore this. And we learned more about it. And we realized that this matrix had a number of special properties, and they ended up being quite therapeutic in the preclinical setting. So then we said, okay, well, how do we fund this? And how do we fund this line of research as using this device or this therapy as a way of either transporting stem cells or used as a standalone? And we said, okay, well, the traditional R01 and R21 pathways were sort of unfavorable at the time to kind of accelerate this forward. So we said, okay, well, let's form a company, and that would give us access to applying for SBIR, STTR dollars. That was kind of the inception. And then in 2021, the aha moment came where we realized that this ECM alone has a therapeutic property on its own. As previously, we just thought it was kind of an inert thing that could be used as a way of transporting cells. And then in 2024, with the help of Catalyze last year, we were able to transfer the production technology to a CDMO biomanufacturing facility to do that tech transfer. And so just to give you some more background. So myocardial infarction affects lots of people, 800,000 Americans a year. Over 25% of these individuals have delayed treatments or repeated heart attacks. And that can lead over the next weeks to months to inflammation, cell death, matrix disruption, as well as progressive heart enlargement and reduced contractility. The grab bag of that is something what we call adverse cardiac remodeling. And that can then lead to the clinical syndrome of congestive heart failure, which affects about 6.5 million Americans, about 700,000 deaths per year. And it is still the number one cause of hospitalization in the Western Hemisphere. And so our matrix derived initially from primary cardiac fibroblasts that were harvested from donor hearts that went unused for transplant. Was the first of its kind natural biomaterial at that time. But we made it another first, which was we took induced pluripotent stem cells, sort of genetically edited cells, which is kind of a big feature in our laboratory, and developed cardiac method to create cardiac fibroblasts, and then use those then to produce the material. So we now have a kind of a cell source that is sort of reproducible and renewable to be and reliable for the production, at least, of this matrix. And this matrix is uniquely composed of fibronectin and other bioactive molecules with well-defined regenerative properties. And the idea was to replace extracellular matrix that's damaged. So we developed an injectable form of this matrix and administered this in a mouse myocardial infarction model. And here's just some slides showing some parameters that many will be familiar with: ejection fraction, cardiac output, stroke volume, using in pressure volume loop assessment as well as echo assessment. And we saw dramatic improvements in the mouse MI model. And we did some immunohistochemistry and noticed that the so-called M2 macrophages, sort of the favorable cell type of the immune system, were upregulated in those mice that developed, that received the therapy. So we think that the mechanism here is in immunomodulation. The production process had been patented through the university. The advantages of this matrix that it's a sort of an off-the-shelf delivery. There's no adverse immune responses anticipated. We've tested this up and down and sideways using in vitro T-cell proliferation assays and a numerous xenogenic treatment models showing no immune clearance problems here. And that's because, of course, this matrix is decellularized. So a lot of the patenting is in how we decellularize it. It is scalable under cGMP conditions now that we know from our work with Catalyze. It's stable for over 6 months and it's readily administered as an injectable. It kind of comes out in the processing as a scaffold, but it can be milled and then particularized into an injectable suspended. And so where did Catalyze come in? Well, it was in this sort of, we had kind of completed 2 SBIR phase 1s. We're not really quite completed. We were in the process of completing them. And then we were thinking about, okay, how do we get this to clinic? What do we need to do to get to the clinic? So that's where the preclinical Catalyze came from. And what we needed through our discussions with the FDA and prior knowledge involved with many other technologies, other industries in the past, is we needed a large animal model. That's what we need. So we need to do a large animal model. We were planning on applying for a phase 2 SBIR. But as we were applying for the phase 2 SBIR, which is about a $3 million award, both direct and indirect, the aspects of the tech transfer and the sort of the production elements, the manufacturing elements, are just not that sexy, though expensive. So they're not sort of things that SBIR phase 2 grants typically like to fund. And so there was just sort of a general interest in funding, the science associated with, sending the matrix into the pigs. In this case, the large animal model would be an MI model in a pig, but the entire tech transfer. So we spoke to Mike Peck, who runs this Catalyze program, and he said, well, it'd be a good idea to kind of get this going to a CDMO. And so we did with Allele Biotech in San Diego, and we worked with them through the entire kind of year and a bit. And we were successful in developing SOPs for production, at least at the GLP level. And we now have sort of the instruments that are needed to get to CGMP when required. Along the way, Catalyze helped us in many different ways, not only in helping to fund that tech transfer, but also supported us through independent stakeholder interviews, things that we sort of put to the side thinking, well, that's not really that exciting. We had done our own stakeholder interviews before, but that's a little biased when it's sort of friends of yours that you include, So this was a nice way of doing an independent stakeholder analysis of, what's the traction of this technology? Where would it fit within normal clinical workflows? What is the cost implications? How would this technology be received in the big societal picture? That's what those stakeholder interviews are for. And in addition, we took a lot of use with RTI's FDA and regulatory submission folks. Mark Levi, in particular, was extremely helpful in helping us kind of think about when the next time we needed to approach the FDA was, because we had already done a pre-IND meeting, but that was long ago when we were using primary cardiac fibroblast, but now we had moved towards induced pluripotent stem cell derived cardiac fibroblast. So that's kind of work that we're still ongoing and working towards that next pre-IND meeting. So why choose Catalyze? Well, a lot of things have already been said, so I'll just kind of gloss over this quickly, but I will tell you there was some financial support for tasks that are ordinarily difficult to fund. There's not sort of support that we got internally to pay for people at our facility in our laboratory, for example, but it did help the CDMO for sure in doing the tech transfer studies. and procedures. They helped us with the stakeholder interviews as mentioned, and they really helped boost our commercialization plan. So we were successful in submitting the phase 2 SBIR, I think, with a really strong commercialization plan, vetted both by NHLBI Catalyze program as well as others. And so we're waiting to hear on the results of that. But, I think our commercialization plan really got boosted there. I'll tell you that the folks at Catalyze, I think everybody is in agreement here, has exceptional expertise. They helped us with the regulatory aspects, the business and commercialization aspects, and even the manufacturing aspects. They individually within RTI and within NHLBI have a wide and deep network, and so we're meeting new people all the time, people that would not normally come through our path perhaps. And as mentioned multiple times, I think now, it kept the CDMO and our team on track for both the timelines and milestones that we needed to achieve. So those are the main features. And I'll tell you that, we have been successful in getting the investor community to really kind of look at as having NHLBI and RTI kind of vetting a lot of what we're doing actually adds a lot of, credence to our approach and gives another eye to those folks to de-risk the technology so that they'd be more willing to invest. So that's in a nutshell. I'll just stop there. Thank you.
Thank you so much, Dr. Raval. That was quite a concise nutshell. So great job explaining your technology and giving us a lot of great points to consider. Our final presenter today is Dr. Vanderslice from the Texas Heart Institute. Dr. Vanderslice kind of bridges the gap, if you will, between the two program arms of the Catalyze program. So the product definition program, which is a grant-funded program, into the preclinical program, which is supported from RTI International, the Catalyze Coordinating Center, and is not a grant-funded program. So Dr. Vanderslice began his Catalyze journey in the product definition program and has now advanced to the preclinical program. So without further ado, Dr. Vanderslice.
Sure. Thanks, Marissa. Get set up here. Can everybody see my slides?
We sure can, yes.
All right, great. As Marissa mentioned, my name is Peter Vanderslice. I'm a senior investigator and director of molecular cardiology at the Texas Heart Institute in the Texas Medical Center here in Houston. My professional career has pretty much been split evenly between the pharmaceutical industry and the more academic setting of the Texas Heart Institute. Now, in my PhD and my postdoc, I was working with enzymes involved with the inflammatory cascade, and that led to a job with a pharmaceutical company developing small molecule inhibitors of a class of receptors called integrins. That are involved in trafficking white blood cells out of the vascular into tissue. So we were developing these for autoimmune diseases. And we were reasonably successful, and ultimately Pfizer bought the company. So a small group of us was recruited over to Texas Heart Institute to use our small molecule drug development expertise to enhance their stem cell therapies they were developing at the time. And fortunately for us, what we found is the same type of receptors, these integrin cell adhesion receptors, would be viable candidates to target for stem cell therapies as well. Now, as you've heard the last few speakers, they've addressed the heart and lung part of NHLBI. I guess mine would be the B part, the blood part here. And the target of ours was the integrin receptor, specifically alpha-4 beta-1, otherwise known as VLA4. And we were trying to develop an activator of this integrin to enhance cord blood transplant. Now, this integrin is expressed highly on hematopoietic stem cells and is involved in trafficking of those stem cells out of the vasculature into home to the bone marrow and then stably engraft. Now, the problem we're addressing is that in most bone marrow transplants, 70% of the patients cannot find a matched donor. Now, umbilical cord blood is an attractive alternative source to bone marrow or peripheral blood as a source of hematopoietic stem cells. And this is due to they require a less restrictive HLA match, which leads to lower incidences of graft versus host, and should in theory make them more amenable to a broader swath of patients. Unfortunately, these preps typically contain 10-fold fewer stem cells than that when you get from mobilized peripheral blood or bone marrow. And so this leads to a slower rate of immune reconstitution. So obviously, once your bone marrow is myeloablated, you want to be able to rapidly reconstitute your immune system with a transplant. Now, the slower the rate of immune reconstitution, of course, the more opportunities for opportunistic infections, results in longer hospital stays and higher costs, and ultimately more graft failures. So our solution, as I mentioned, is to develop an activator of this integrin alpha-4 beta-1, or VLA4, to promote the homing of the stem cells into the bone marrow and leading to stable engraftment. Now, in theory, this should correlate with an acceleration of the time to immune reconstitution. Now, I should point out, we didn't start from scratch. We did have an initial lead compound, or what I'll call a parent compound, that did activate this integrin that we had developed. We had some pilot data showing some efficacy in a preclinical animal model of cord blood transplant. But this compound really was not ready for prime time in the context of this indication for two reasons. We were trying to develop an orally available compound, and that parent compound had very low oral availability. More importantly, it also was metabolized and was an inhibitor of cytochrome P450 3A4, which is an enzyme in the liver that metabolizes a lot of drugs. And we were concerned with drug-drug interactions using that compound in combination with other drugs that are typically used in this setting. So those were the two major issues that we wanted to improve upon our parent compound. So I thought it'd be useful to show you what the milestones and metrics for progression were, because all of you will be required to do this if you write a Catalyze grant. And they revolve around milestone 1 is developing compounds that would be less reactive with CYP3A4. Milestone 2 involves developing a more orally available compound. And then the rest of the milestones involved with the preclinical animal model. So our chemists were charged with synthesizing at least 10 compounds per quarter. These compounds needed to be at least within 2-fold activity of the parent compound. And those that were then tested in our CYP3A4 in vitro assay. And this was the high hurdle to get a better than 50-fold improvement in 3A4 activity, meaning 50-fold less active. And only then would it go into an oral pharmacokinetic study in mice. And there, the metric was needed to be 10-fold higher than the parent compound. And only then would it go into our cord blood model. And the metric there was, although the parent compound isn't orally active in this model, we wanted our compound to be orally active in this model to the same extent that the parent compound was IP. And then it would go into a long-term engraftment model to show that it wasn't, it was keeping the multi-lineage reconstitution intact. From all this work, we identified a compound 7-HPre-935, and the metrics are shown here. Relative to the parent compound, it was equipotent in VLA4 activity. It had a 70-fold improvement in 3A4 activity. And the mouse pharmacokinetics is shown in the lower left, and the red is the parent compound, and the blue is 935. And the y-axis is in log scale. So it's a big improvement in oral PK. It's 20-fold higher AUC, area under the curve, and a 38-fold increase in Cmax. Additionally, we're interested in lowering the molecular weight of the compound to less than 500 and log P less than 5. So any chemists in the audience will recognize that these are consistent with the Lipinski's rule of 5 for developing an orally available compound. So then we put it into a xenogenetic cord blood transplant model. This was just a 2-week model to screen any compounds. And so this model involves using human CD34 positive umbilical cord blood cells transplanted into a non-skid mouse. And then we harvest the bone marrow and peripheral blood at 2 weeks and measure the percent of human CD34 cells in the bone marrow. And you can see with compound treatment versus vehicle, we get a 3-fold increase in stem cells and grafting, which is what our hypothesis was. And fortunately, this did correlate to an improvement in human CD45 leukocytes in the blood. So a 2.3-fold increase there with compound. Now, 2-fold may not sound like much, but to a transplant doc, if you can cut the time to immune reconstitution in half, that would be huge. We have since put this compound into multi-lineage reconstitution, and we found that, you know, at the early time points, it does show this improvement, and then ultimately it does not, mess up the multi-lineage reconstitution. That's all kept intact. So the impact of the Catalyze Award, I've sort of separated into tangibles and intangibles. The bottom line is we couldn't have been able to do this without the Catalyze funding. It just, most academic labs are not set up for this. But the tangibles include, we got new intellectual property, in this case, composition of matter for the class of compounds. We have out-licensed that to an accelerator partner. And all this data allowed for us to write and be awarded future grant support for IND enabling studies, so investigation new drug enabling studies for the FDA. This includes a fairly large CPRIT award, which is the Cancer Prevention and Research Institute of Texas, which is a fund set up in Texas to fund cancer research, obviously. We also were fortunate enough to get, as Marissa implied, from NHLBI, a Catalyze preclinical services contract, which again, those funds don't go to our institute, but they do a lot for moving your compound through that valley of death, going through toxicology, metabolism, and et cetera. Also, a side benefit to this grant was it supplied matching funds for our CPRIT award, which was a requirement for that award. One particular thing that's been useful lately is updating our commercialization plan. Now, our original plan was targeted to umbilical cord transplant, but sort of a bigger patient market is more the broader allogeneic transplant patients. And so we're still going to focus on cord blood as well, but we've modified our commercialization plan and are developing it for both indications. And it's the Catalyze has been very helpful in that regard. In terms of intangibles, Catalyze funding allowed for a focused, concerted effort to achieve this goal. I mean, academics a lot of times is characterized by sort of every man for himself, where this really allowed us to be a true team effort to achieve the goal. And you've heard this from the other speakers before. I sort of view it as the meetings are sort of run like a pharma industry team, where you have quarterly meetings, milestones, and metrics for progression. And so this arrangement, we were very comfortable with this arrangement for moving the project forward based on our past experience. And the annual meetings are very worthwhile. Personally, I found the biggest benefit was from the workshops they do on pitches. And these are very interactive workshops, I should say, on sales pitches, target product profile, product development plans, and interaction with the FDA. And that's just a few of the topics, but I felt those workshops were really worthwhile. So I appreciate you guys spending the time, and I'd be happy to answer any questions.
Awesome. Thank you so much, Dr. Vanderslice. All right. So as promised in our agenda, we've gotten through our 4 investigator presentations. Now we'd like to take your questions, and specifically your questions for our investigators. So I'm seeing a couple, and please continue to type your questions into the Q&A that you'll find at the bottom of your Zoom screen. But first, we have, what are some of the key differences in your experience between a milestone-driven program like Catalyze and maybe other sources of funding like an R01 or any other traditional sources of funding?
Marissa, I can answer that question. Initially I was a little intimidated by a milestone related grant, but as I moved through the process, I really began to appreciate the value of that type of grant in that, one, it really kept me on track. And allowed me to move my internal meetings along well, recognizing that we had milestones to keep, but also the milestone meetings themselves, I think, were particularly important, as I mentioned earlier, because you were able to engage with some very thoughtful leaders who allowed you to identify some potential barriers and how to get around those barriers. So both the keeping on track, allowing me to learn how to pace myself more, and also being able to get frequent feedback with the milestone meetings, I think were very valuable to me.
Maybe I can add a little bit from my perspective, the preclinical pathway, as mentioned previously, we had already received and have been working on 2 SBIR phase 1 studies that are also sort of milestone based. But, those types of grant mechanisms, typically the interaction with the NHLBI is one way. We provide a progress report and either, say, okay, thumbs up, keep going, or not, or, there's not a lot of feedback. But this is different. This is, regularized meetings, verbal back and forth, very interactive. If we're kind of, and it's a much more, I think it's because it's real time. So you're not sort of going down tangents or thinking about prioritizing things that really ought not to be prioritized. So I think, I know the question is more about, how does this differ to other sort of traditional hypothesis generating, type grants, that we're trying to answer a hypothesis. Here, this is much more development, but compared to other development grants, this is much better because it does have that human touch and that more greater degree of interaction.
Thank you so much. Any other comments from panelists?
I would just add a small thing. It's easy to get distracted as everybody's running their labs, but the quarterly meetings were very useful for keeping you on track, like I said. And sometimes you dread that email where they're scheduling it, but and it got everybody together and you kept the ball rolling. So that was much appreciated. And also, as the other speakers have mentioned, it was a very interactive meeting. It wasn't just like reading a book report or something.
And I'll add that I think overall for us, it helped de-risk some of the process. As most of us engage in research, things go in different directions or don't work like you expect them to. So rather than waiting, to the end of the year or trying to find other folks to give input, having folks that are familiar with the process and your project and what your ultimate goal is, really helped us with some key decision-making process that we had to pivot a little bit in the middle. So that was probably the main takeaway for me.
Great, thank you all. I think the next question I will ask Dr. Gundry to start. So this is from an audience member who says, I heard about Catalyze looking at info about the R61, R33 opportunity. Did any of you approach Catalyze by this path? And Dr. Gundry, I know that you did. So maybe I'll expand on this question a little bit and ask what your experience with the biphasic award was. Maybe anything unexpected that you encountered, or just kind of the process of that award.
Yeah, so we had the R33, which is the kind of the, I guess conceptually maybe the earlier one. Really just gave us the freedom to just start. A lot of it was from scratch. So here's this product. We think it's going to be a product. And we had to go through a lot of, just the early phases of moving it from just a research tool to something that could be commercialized. So we haven't done the 61 part yet. But I think that we learned so much and really positioned ourselves to be prepared for the next step for sure. And that's, benefit of all the activities that we had throughout the program. But I started with the R33. Yeah.
Oh, sorry, I am mixing up my my awardees. Dr. Benza, I believe you have a biphasic award. Would you want to take a stab at that question?
Yeah, I thought this was a great opportunity to move from a concept that was really on a piece of paper to an actual product in the R33 phase, and then using that prototype that we developed, learning a little bit about the regulatory and commercialization in that first feature, and then moving to the R61, where we're able to do preclinical studies with the prototypes that we developed in the R33. And that's where we are right now, is doing preclinical trials in animals with the catheter, really showing that it works. It's really been very rewarding to participate in that biphasic process, because it just brings you through all the steps naturally. And I know with my next concept idea, I really have a great background now going through the biphasic process and how to move a product from my mind all the way to the preclinical trials. It's been a really great experience for me.
Maybe I'll jump in with a follow-up question. So traditionally with NIH grants, you don't want interdependent aims. This is one of the questions and something we get a lot. So maybe for the folks thinking about grantsmanship, especially you, Ray, what was your process through developing the R61 versus the R33 like milestones?
So in my mind, the R33 was really structured around developing a working prototype that I can bring to a preclinical model, and then designing the preclinical model that replicates the clinical condition as much as possible was also kind of a challenging but exciting part of the project too, because it's really the only way to test in bed the versatility of the product you're designing in my type of industry. So it was again, it was a very good learning experience.
Thanks for that. I'm not sure if we can get people off mic, but I'm seeing a lot of questions regarding process and basic information about the program. And just a reminder, we have a full FAQ page on the website, as well as webinars related to how. So maybe back to Rebekah. I know you have some other types of more traditional grant funding mechanisms. I'm just wondering, now that you've gone through Catalyze, how has that influenced your other types of research within your academic laboratory?
So I think in a couple of ways. One, I'm an analytical chemist. I study the heart. It's always a battle to convince reviewers that in a traditional mechanism of, hypothesis driven, because we do so much tech development. And so this is a mechanism that actually fits a hole that we can't get from kind of the traditional purely hypothesis driven. They don't want to generally, I've never met a reviewer who wants to see my discovery, right? They're assuming I've already done the discovery, don't propose discovery, and don't propose aims that are dependent on each other. This gave us really the freedom to actually do the science we needed to do without those kind of conceptual constraints. So we had, great proof of concept, but we were very much structured on, the aims were really milestones in that, but we did try to separate out milestones that would be not completely dependent on each other. Although when you're doing product development, that's kind of hard. But I think that this mechanism gave us the freedom to that, and really was the right thing for this technology. And it also helps me now going forward, really being able to better parse out some of our projects and delineating between the things that are really product or commercialization sorts of things from really those hypothesis-driven biological studies. And so I think that's how it's driven us. And like I also mentioned, it's opened the door to really considering other business-based grants that I don't think I ever would have thought about if I had not gone through this program. So for me, it just opened kind of the door, and probably really just in what I felt comfortable approaching. And now I feel like we're better equipped to be competitive at those sorts of applications than I would have been before.
Thanks for that. So just to clarify, the specific aims are what's peer reviewed, and those are your broad overarching goals of the program. But then for Catalyze specifically, we have milestones which break down those big aims into discrete steps that we can then track. So the next question, I know several of you had to pivot throughout the program. So I'll leave it up to whoever wants to answer. What was the key determinant in changing a pivot from a milestone that we had negotiated beforehand, and kind of the thought process between changing the milestone from the originally agreed upon approach?
I can start. So from our experience, this was the great part about the fact that it was a two-way communication. So when we had our, milestone meetings, we could bring to the table, hey, this is what we proposed. Here's what we tried. We've tried these 5 other things. And this is why we think changing, this technical aspect to this is actually where we need to go. And we just had, a really good conversation back and forth about, yeah, that actually makes sense. And I don't know that it's totally different than how you would do a normal grant, as in, you follow the data. But here, because it's milestone driven, it was just a more structured approach. But it was still, you still had that intellectual academic freedom to follow where your data were taking you, because you still have that same ultimate goal of making a product or a drug or something that is going to achieve your end goal. And you have to be a good scientist in that path.
I could perhaps speak to, as I said, we had like a big CYP3A4 aim and a oral PK were two of the milestones. And initially we had lots of compounds that were greater than 30-fold, but not greater than 50-fold for the CYP3A. And so we were getting anxious to move more compounds into oral PK than we were able to do based on the metrics. So we discussed, lowering that aim to 30-fold just as a metric for progression into the oral PK, which mutually the team decided that was okay. So we did that. And so we got more compounds into PK. Now, ultimately, we wound up with one of our earliest compounds actually was 935, which was 50-fold better. But other than that compound, we were having struggles initially getting past that 3A4 aim. But it was just within discussion with the Catalyze team, we lowered that to 30, just so we could see more compounds in the PK, because sometimes you run PK, not that this is the golden compound, but it has some structural change that may be useful to know how that's impacting PK. And so it allowed us to get a much more better analysis of what was required orally by lowering that aim, that metric. But ultimately we wound up being able to stick with the original metrics at the end, but that's just an example.
Great. Marissa, I think we're at time, so I'll hand it back to you.